Hello everyone, my name is Ashley Grover.
My name is Alberto Herrera.
My name is Ashley Cotopozzo.
And I'm Andy Boniface.
And we worked in the mechanical engineering department with our mentor Christakis Andreou, and our principal investigator is Professor Paul Meinhardt. The focus of our project is mixing studies into microfluidic devices. So microfluidics is generally a newer branch of technology. It deals with behavior, precise control, and manipulation of fluids on the microscale. And to get an idea of how small the microscale is, here's an example of a microfluidic device. The channels on this device have a diameter of approximately 100 microns, which is just about the same diameter as a single strand of hair. Microfluidics is very efficient in that we can perform lab tests very quickly, and it costs much less than lab-based methods.
So microfluidic devices allow scientists to very precisely and accurately study and analyze what is happening with particles on the nanoscale level. This is a device that our mentor, he built it and developed it. And this device will allow him to analyze what's going on past the junction point. And there will be diffusion going on further through here, but he'll be analyzing the detection of drugs, and he'll be able to look at these particles in the microfluidic device to see what is going on. And this is a closer, up-close picture of the chemicals, or the fluids will be mixing as they go further through the device. And as he controls the flow, he will be able to control when the chemical reaction occurs, and that will let him see all of these particles mixing. So beginning the project, our goals were to build a microfluidic device, and we wanted to operate it and with constant pressure flow. With the constant pressure flow going, we wanted to put fluorescent dye in it, and we could see what is happening on the nanoscale level. We will be doing this, we did this with microscopy techniques, and we were looking through the microscope, taking pictures, and we can analyze those, look at those, and see what is going on with the laminar flow conditions and the droplet conditions.
So this is the geometry of what our device actually looks like. As you can see, there's 4 inlets on the left side, and there's 1 outlet reservoir on the outside. The fluids come in on the inlets, and the pressure pump is placed on the outlet. The pressure pulls all of the liquids, and they combine in this one junction. To build a microfluidic device, we start with a prefabricated PDMS mold, and we apply a UV curable polymer that cures for 7 minutes curing meaning it dries from a liquid to a stiff solid plastic. After that, we carefully peel the PDMS mold off, and then we have the we have the device with the structures in it. We then repeat the process with a polydimethyl siloxane mold, but this time it's flat, and it's only this is to create a lid that will go on the top part of the other mold. This one cures for 4 minutes. This time to make it to make it hard enough so that it creates the lid, but not too hard so it can still stick to the rest of it. We peel it off the PDMS mold, and then we have our device with the channels going through it. This is a blown up picture of what our device actually looks like. And here you can see how the 2 glass slides sandwich the 2 plastic slides in between it, one with the actual channels flowing through it.
So fluorescent dye and water are injected into the device through the inlets. A vacuum pump is connected to the outlet and pulls the fluids through the device. And the fluids, they're moving at such a high velocity, and the particles are so small that this causes, this allows for laminar flow. And in the laminar flow, the fluids flow in smooth lines parallel to each other, and no mixing occurs. But as we can see in this magnified view of the channel, we can easily distinguish the different fluids that went through the channel. The three white flows are the fluorescent dye, and the two darker ones are the water.
So there will be some circumstances where a slow type of mixing will be preferred. In this case, our microfluidic device can utilize slow mixing by something called diffusion. Diffusion is the movement of particles from an area of high concentration to an area of low concentration. And to get a picture for what diffusion looks like, imagine dropping a few drops of milk like in a cup of coffee. You'll see that it starts to disperse, and that's what diffusion looks like. So in this case, we'll have a vacuum pump on the outlet pulling all of these fluids in together, and they will all come in contact at this end over here, and then they'll all run over here. Now, when you have the different fluids running through, you'll have the laminar flow appear, but the laminar flow will eventually start to diffuse, as all laminar flows do. Depending on the speed of the velocity of the particles will determine how fast they diffuse. So the lower the velocity, the faster they diffuse, and the higher the velocity, the slower they diffuse. And this allows us to have complete control over how fast or how slow we want our particles to mix. So you can also
By measuring the distance that the molecules travel and the time that it takes to do so, you can use Einstein-Stokes equation and Fick's law of diffusion to calculate the approximate size of the molecule used, in this case rhodamine, which is the fluorescent dye particle that we used. We calculated it to be about 20 nanometers, but in reality it's actually 1 nanometer. This difference is due to the magnitude of the scale. Since these measurements are so small, any minor mistake can cause this difference.
So we can also have fast mixing with droplets. And what we did here in this experiment is we added cyclohexane in these two channels, which is a type of oil. We added fluorescent dye here, fluorescent particles here, and water. So when we add the vacuum pump and pull all of these liquids together so that they come into contact and start flowing, we get these droplets over here. Now, because oil and water do not mix, the oil runs on the edges of the channel, while the water will only want to run in the center. And because the water will only want to cling on itself because it doesn't want to mix with the oil, they will form spheres that resemble droplets. In these droplets, you can see that half of it is really bright and the other half of it is really dim. The bright one is the fluorescent dye, and then the dim one is the fluorescent particles. The dye emits its own light, the particles do not. And you'll see that in the beginning, they're clearly separated. But once they run over here, by this end, you'll see that they become a little more mixed. And this is how we do fast mixing by the droplet process.
So throughout the whole project, we were able to build a microfluidic device. We were able to run experiments and test experiments and see how the flow works throughout the microfluidic device. We were able to see how the flow through the channels go, and we were able to understand diffusion. And after doing that, we got to take pictures, and then we were able to measure as precise as possible how big our results were. And then doing that, we were able to apply our results to equations and try to get an answer for the size of a particle. And we weren't completely successful, but we got the idea of it.
We would like to give a special thanks to our mentor, Christophus Andrew, and our PI, Carl Meinhardt. Also, all the program staff, Ophelia, Anthony, Jens, and Jacob. Also, our RAs, especially Myra, thank you very much. The University of Santa Barbara, the California Nanosystems Institute, and the Center for Science and Engineering Partnerships. Also, the Jack Kent Cooke Foundation.